everyone hear me okay? Or should I move my mic up a little closer? Good, awesome. Let me know, stop me if you guys can't hear me well, and I can always adjust the mic to make it easier. Just want to start off by thanking everyone. Oh, perfect, awesome.
Hey guys, thanks for coming. And first of all, I would like to express our huge gratitude to this company, Genomics, who is supporting us in very many different ways, including this giving us basically free reagents, which allow us to produce some of these workshops where basically you guys can do small pilot projects for free. We have done one a couple years ago where we actually introduced the first version of this Flex kit. Back then it was also a small pilot where everybody would get 4 samples, which should be enough to generate some preliminary data, and the idea is that this data can be then used for bigger applications. So this time we're moving to the new version of the Flex kit, which is called Genomics Flex, which has certain advantages, which I'm sure Juliana will talk about. And again, this is sort of between this cycle, so this is all came rather unexpectedly, but there is this announcement of 2 pilot projects will be awarded to the applicants, where you will have 4 samples to be submitted. And the beauty of this Flex kit is that it allows you to work not only with the fresh cells, but with fixed cells, with frozen tissue, which can be what's called chop fixed into single cell suspension fixed nuclei, and even FFPE samples can be worked. There are still some limitations. The minimum number of cells is still rather large, even though it became much smaller than what used to be in the first version of this kit. All right, with that, I don't want to take too much from Juliana. Juliana Crivello is our field application scientist. She will be presenting today. Again, huge support from these guys. They have office hours for us. They often provide these free reagents, and also thanks to GSI, who fund our labor from the single cell core facility, and also to Illumina, who happen to provide free sequencing kits. So it's basically almost free even though this time there is a small copay under $1,000 for a project that would otherwise cost more than $10,000. All right, so Juliana, all yours. Also, Alisa and Lauren are here, other representatives from the company.
Hi, everybody. Thank you guys all for coming today. And so like Yuri said, I am your single cell field application scientist. So as you get into your 10x experiments outside of events like today, I'm here to help you guys troubleshoot, help with training. Like Yuri mentioned, we have Elisa and Lauren in the back. They're part of the pre-sales team. So if you have questions on pricing or ordering your own kits, those would be your go-tos for those types of questions as well. And with that, we're going to go ahead and get started. So the goal of today is really a super condensed crash course in what Flex data looks like and how you guys can use some of the tools provided to you by 10x to dig into your Flex data. Obviously, with the intention here that you guys can come up with ideas for your own grant proposals for the competitions that's going on through the GSI and the sequencing core. So with that, I'd like to get started by just kind of doing a primer for what Flex is. So we'll touch on everything at a high level and in terms of the major steps of the workflow and how we're going to generate the data. And then we'll switch gears and we'll dive into some data specifically for today and for you guys here at BU Med. I have picked some chop fix data, which I think is going to be really relevant to the tissue types and sample types that Yuri and myself have seen from a lot of the researchers here on campus. So I went through and I analyzed that data using our fresh version of Cell Ranger so that you guys can get a real feel for what it will look like when or if you were to win this great proposal and have your own data to process as well. So with that intro, let's go ahead and get started and just talk about what Flex is. So for really any of our single cell assays, we have these 4 major steps that we can break the workflow up into. And first, right, we have sample input for our traditional reverse transcription-based capture methods. This is typically where we spend a lot of time doing optimization, right? Having really high viability samples leads to high quality data when we're talking about our RT capture assays. Here, when we're talking about our flex assays, we're talking about fixing your samples. There are a lot of different types of sample inputs that you can use that are compatible with our fixation protocols. So I'm just going to highlight what those protocols are on the next couple slides. Right, so if for whatever reason you are interested in working with nuclei specifically, you could isolate nuclei from your frozen or fresh tissue using our nuclei isolation kit ahead of fixation. Alternatively, and what I think is one of the coolest options with Flex is you can actually take scrolls from your FFPE blocks and isolate nuclei from those scrolls. So I feel like most researchers, right, have that FFPE block or a set of FFPE blocks that's been kicking around in the lab for years. And as far as molecular, there's not a lot of different experiment types that you can do with that tissue in that block. But now we've really changed the game with this isolation protocol. It's incredibly straightforward to work with. The biggest caveat there is the quality of the tissue prior to being embedded in that block. Next up, if you're working with whole cells or frozen or fresh tissue where you don't want to do a nuclei isolation, there are a couple different protocol options for you here as well. Obviously, anytime we have cells or nuclei in suspension, we can go ahead and just fix those in suspension and lock their RNA states in place. If we're going from fresh or frozen tissue, like Yuri mentioned at the beginning, we have what we call the chop fix protocol. I really love this option. I wish this was around when I was still a customer dealing with tissue samples. For most people who are getting frozen tissue, traditionally you've been sort of boxed into doing a nuclei isolation ahead of your RT capture for your single cell experiments. Here with the chop fix protocol, what you'll do is you'll take that frozen tissue, you'll chop it up into little pieces, you'll fix those pieces, and then do a dissociation. The benefits with this protocol is that you're more likely to retain your fragile cell types in that final cell suspension that you're going to have going into the single cell library, right? And this makes sense intuitively. We know that some of the dissociation methods we use even on fresh tissue are harsh, and those fragile cell types are going to lyse during that dissociation. So chop fix is a great alternative to some of those methods in order for you to capture the full heterogeneity of your sample. And then lastly, while it's a little bit more geared towards clinical trials or big clinical studies, we do have a whole blood fixation protocol. So this is particularly handy if you're collecting blood off-site somewhere and then transporting it or shipping back to the lab to do the downstream processing. Essentially here what you do is you add the fixative buffers directly to the whole blood. This allows you to ship at 4C overnight, and then you can do the downstream processing, PBMC isolation, whatever it is that you want to do prior to putting those cells on the chip. Like Yuri mentioned, as potential winners of this grant, your responsibility will be to fix your own samples and hand them over to Yuri's core for that downstream single cell library prep. And he also mentioned, the caveat around numbers. There will be some wash and spin steps that Yuri's core will have to do with your sample prior to getting it sequenced. And so to that end, we generally recommend 300 to 500,000 cells going into the hybridization reaction that Yuri's core will be performing. So that's sort of the landing zone of cells that you want. Ideally, we want more than that, so that way we can give Yuri's core plenty to go into that hybridization reaction. And like we mentioned at the beginning, I do have office hours. If you're struggling with yields or you're not really sure how to prepare your sample, I'm happy to meet with you guys and talk about it in more context of your project and the constraints of how you're collecting your samples, so that way we can make it make sense for you guys as well.
Juliana?
Yeah.
So what's the absolute minimum that we can go with?
Yeah, I mean, the absolute minimum that we say online for GemX Flex is 25,000 going into the hybridization. But it's really tight, and it will really be dependent on how well those cells will pellet, right? Because we just know there's going to be inherent loss during the wash spin steps. So I would feel probably most comfortable with like 100,000 going into the hive. I think that would be like a conservative hedging our bets number. Okay, so like I said, happy to meet with you guys individually. I know I do have office hours this Friday. All of my time slots have been booked up. If you would still like to meet with me, you can reach out to me via email and we can schedule a separate time to talk about your project and your samples, so that way we can sort out the best protocol for you to use. Okay, So the take home from the sample input, right, there are a lot of different sample input types that you can use for Flex. It's one of the reasons that I like Flex. It gives you a lot of flexibility in how you can do the downstream single cell library prep. And once we prepare our samples and hand them over to Yuri's core, they'll be responsible for these probe hybridization and gem generation steps, as well as the downstream library prep and sequencing. So let's first talk about this probe hybridization, right? So once we've got our cells fixed and permeabilized, regardless of the method that we do them, we hand them over to Yuri's core. He's going to incubate our cells with a whole transcriptome probe set, either human or mouse. And you can see here we have about 18,000 human or mouse protein coding genes that we're targeting. If we look at the schematic over here on the left, you can see the overall probe design for the probes that are targeting those 18,000 different genes. So we use this probe pair combination. So we have a left-hand side and a right-hand side probe. There's going to be 25 nucleotides of complementarity to the gene of interest for both of those. So the intention here is that these two sides of the probe will come down and hybridize to their mRNA targets directly adjacent to one another. This allows us to then ligate these probe pairs together in a downstream incubation step. So other things to note about these probe pairs, depending on the relative gene expression of the particular gene that we're targeting, there could be anywhere from 3 to 10 probe pairs that are targeting the body of that gene. Why am I mentioning this? One of the benefits of Flex, because we've done this, so for our really high expressing genes, we'll have, let's say, 2 or 3 probes targeting the body of that gene. For our really low expressing genes, we might have closer to 10. The reason we've done this is this allows us to sequence to a shallower sequencing depth, so we don't have to sequence as much to capture the full complexity of your library. So not only are there cost savings on the multiplexing, which I'm going to talk about in a minute, but there's also cost savings on the sequencing end as well, because we made it easier for you to sequence the full complexity of these libraries. Okay, so we've talked about how we're saving on sequencing costs using this probe-based system. The other thing I want to mention is how we're saving on the number of samples and the number of cells we can get in a given library. And that's really reliant on what we refer to as our probe barcode that you can see here highlighted in gray. So for our probe barcodes, we have 16 for the most recent version of Genomics Flex. We're currently launching a version 2, which is actually going to have 384 different probe barcodes, but for now, with the version we're using for this pilot program, there are 16 different barcodes that can be found here. So as you can see in this little schematic down here in the bottom, what we have is you would receive 4 different tubes of those human or mouse whole transcriptome probes, and each one of those tubes would have a different barcode at this location for all of the probes within the tube, right? So we're still targeting all 18,000 genes, and across those probes within a given tube, they're sharing this sequence. So what we'll do is we'll take that tube of barcoded probes and we'll hybridize it with one of your samples. We'll take the next tube that has a different probe barcode and hybridize it with a second sample, and so on and so forth until we have 4 unique probe barcodes that are individually being hybridized to 4 different samples. So we'll allow this hybridization to occur overnight, and then once that hybridization is done, we can now pool the cells from all of our different samples because we have essentially a sample barcode for each one of them, and then we can process them together as one downstream single cell library. This is really where the money savings is coming from. It's allowing you to flex more samples into one library and still get a good coverage of cells per sample on the out.
Juliana,
Yeah.
but for this grant, however, it is a single barcode.
It's not going to be a 4-plex. I thought it, well, okay. Well, this is, so for the pilot, you guys can get your data, and then in the future, the cost savings are there for you guys to flex it and drive down the cost for a future study. So for this pilot program, you guys will be generating 4 separate libraries for 4 separate samples, but just keep in mind for future studies that you have this multiplexing option available to you. Okay. So we've talked about how we're hybridizing the probes to our samples, how we have the option for multiplexing down the road. Yuri's core will be responsible for generating that single cell partitioning, which will happen in droplets on their Chromium instrument. And then we will go into library construction and sequencing. For library construction, once we get our cells into droplets, the first thing that we'll do is we'll ligate those hybridized probe pairs to one another. We'll release them from their mRNA targets, and then we'll pick up these probe pairs using an adapter sequence on this 3' end here that's going to be complementary to the oligos on the gel beads that are encapsulated with our cells. And so upstream of this adapter sequence, we're going to have our 10x barcode or our single cell barcode. So any molecules captured by the oligos in this gel bead inside this droplet will all share that 10x barcode, which is why we refer to it as a cell barcode. For people who are familiar with our assay, we do have something called a multiplet rate, which is just the statistical likelihood of encapsulating one or more cells into one of these droplets. When you get into multiplexing, one of the benefits here is that we have these probe barcodes, so we can actually differentiate when there are multiple cells in the same droplet, and you get to retain a little bit more of your final data. Once we pick up that adapter sequence and incorporate our cell barcodes, we have this fully extended ligated product. Yeah, Nelson. Oh, sorry, couldn't see over there. Yeah.
How efficient these barcodes are to detect mutations in the transcript?
So, are you talking like a single nucleotide?
In the parent transcript.
Are you talking like a single nucleotide polymorphism? Yeah, so you would have to go, so all of the probe sequences are publicly available on our website, so you can actually go in by gene and look at specifically where we're targeting. I will say for the left and right hand probe pairs, single nucleotide changes would need to be within 4 bases of the junction site for it to be sensitive enough to pick up that nucleotide change. So generally we tell people it's not sensitive enough to capture SNPs. There's the option for designing custom probes, but again, you would need to be really careful with the design there. I think as far as mutations go, things like deletions or alternative splice sites, those tend to be a little bit easier to design custom probes for because it's a little bit more cut and dry, and you can kind of design the left and right hand probe accordingly. Yeah, but good question. Okay. So we will extend our products right at the end of this incubation step where we're incorporating these barcodes. We'll have adapter sequences on either end of our products. Everything will be single stranded. So the first thing we'll do once we get out of our droplets is just amplify everything up, make it double stranded, make a bunch of copies of everything. So that way we have enough material to go into library construction. Yeah, Nelson.
Another question about target capture. Are there instances where there's a gene that's so abundant that it soaks up all of the probes and then it plateaus or maxes out?
I see what you're saying. So it's being so highly expressed that there's just no probes left for the rest of the cells in that sample. Is that? Yeah. I haven't seen that in any of the samples. I will say, just based off of what I've seen, like on traces when we go to clean up from the PCRs, the oligos are in such excess of the sample that I don't know if that I've seen a saturation event like that, just because typically we have a lot of unbound leftover probe, which is why we have all of those probe post-hybridization wash steps, is to get rid of all of the unbound. So it's in such excess, I haven't seen that.
Well, I can imagine the bulk of all the probes is in excess, but if there's a specific gene, it could theoretically eat up just those specific probes of that gene. Like you see every plateau, is there like a...
Yeah, I haven't seen it in the data. I see what you're saying, but again, I just don't know in general, considering I've seen like just how much leftover probe there is there. I would be surprised if that was possible. Yeah. Okay. So we are going to amplify up these products. We have enough material for library construction, and then library construction is going to be much more simple and straightforward than a typical transcriptome library prep where we're capturing the full transcript. Here we're just capturing the 50 nucleotides of our probe pair and sequencing it. So really all we need to do to make these libraries is to incorporate our sample indices for that paired end sequencing. So we'll rely on our adapter sequences to incorporate I5 and I7, and P5 and P7 will physically bind us to that aluminum flow cell for sequencing. Okay, talked about how we're fixing our samples. We talked about how these probes have been designed to hybridize to your transcripts in your fixed samples, how we're going to make those libraries, and we've talked about how they'll then be sequenced using paired end sequencing. So now I want to switch gears and just talk about what the data analysis looks like. To start, I'm just going to run you guys through our Cell Ranger algorithm at a high level. Cell Ranger is the pipeline that's used for processing any of our single cell data that's generated through 10x. And so for Flex specifically, here I have an example of multiplex Flex. So you could just remove this sample assignment for your single plex pilot experiments. But But essentially, once we sequence your libraries and we pipe them into our Cell Ranger algorithm as FASTQ files, the first step is just going to be aligning, right? And here what we're doing is we're aligning those sequenced pro pairs, that 50 nucleotide sequence that was hybridized to your transcripts. We're aligning it to our probe reference, right? We have a CSV reference file that contains all of the sequences of all of the probes that are known to be there, and we'll align to that as well as the transcriptome. We'll then take the band output file from that alignment and we'll do our counting algorithm. And here, really what we're looking at is differentiating background noise and empty droplets from true cells. And so we'll count the number of, oh, excuse me, I'm out of order here. So we'll count the number of features. So here we're just looking at the number of transcripts or really probe pairs that we're capturing. We'll take that raw file, and if you're multiplexing, then we'll do a demultiplex based off of those probe barcode sequences. For the pilot, this isn't something that you guys will need to worry about. And then we'll go into calling cells for each individual sample in the multiplex library. And so here we're differentiating between background noise and true signal of each individual sample, making sure that your final data is only going to be relevant biological information. Lastly, for the Cell Ranger pipeline, there's going to be this secondary analysis where we generate these cluster plots, differential expression analysis, and a couple different outputs that I'm going to highlight in the next few minutes. Any questions on the overview before we switch gears and talk about what the output files from Cell Ranger look like? Excellent. Okay. So I've tried to put this together a little bit cooking class style. So I have previously already run some data through the cloud. So what I want to show you guys first is just how to access the cloud and how to download your files from the cloud once you've run it. So here I'm already logged into my cloud account. Any customer can sign up for an account for free on our cloud website. I think there are limits on the number of analyses that you can do on a given data set, but you could always re-upload that data set into a new project and keep analyzing it. So there are ways to use the cloud for free, definitely. Let me zoom in here. So this is sort of the home landing page. If you're an instrument owner, this is where you'll manage the instruments that you are tied to. And this is also where you're going to get access to our cloud-based version of Cell Ranger. So if you're not a bioinformatician like me and you're not comfortable, being in a command line environment, the cloud is a really great option for you to process your data without having to be able to write scripts or execute things in the command line environment. Okay, so I'm going to just go to my cloud analysis page, and I have a project that I already created a few weeks ago ahead of this event. Like I said, what I've done is I've just taken some public data off of our website, and this was a 4-plex, so it's a multiplex flex library where our R&D team used mouse eye, ovary, intestine, and stomach tissue and flexed those 4 tissues together in a single library. So I thought this would just be a nice highlight to highlight different tissue types that can be used with the chop-fix protocol and sort of what to expect for the data output. So like I said, I've already run an analysis on this data. So I'm just going to click over to the Analyses tab. And I just did a quick default parameters, nothing fancy. I just want to see what the data looks like. So once that analysis is complete, there are a couple different things that you can do within the cloud. So I can automatically click right here and open a QC report. Or I can go down and look at all of the different output files from that Cell Ranger run. Anytime we're doing a multiplex flex run, there's going to be like an overall library output file, and then there's going to be individual output files that are going to be on a per sample basis. So I'm just going to focus on the per sample outputs for today. If you're interested in the whole library, you can always go into this top folder and pull out those whole library raw files. So for each individual sample, we can see here there's its own folder. And then within that, we have the summary HTML file, which I'm going to show you guys what those look like in a couple of minutes here. But these are going to be essentially your first pass for just seeing what the data quality looks like, what your sample quality, a lot of QC metrics in the web summary. So it's really helpful to understand how to interpret. And then we have this count outputs folder. This is where all of those secondary analysis files are going to be found. So like we talked about, there's going to be some feature barcode files, a lot of file types that can be piped into things like R for doing, your tertiary analyses, generating figures for your paper. Today, what I'm going to focus on are these Loop Browser files. So Loop Browser is a free data visualization software off of our website. I like to joke with customers that I think it's also for the non-bioinformatician, the bench scientist. Again, similar to the cloud, you can click around and play and look at your data and start to get some biological insights. Especially, I find sometimes when you're working with a bioinformatician, you don't always know what it is that you're looking for. You just kind of want to get a feel for your data. And then once you kind of see what the cell types are, or what the genes are that are being expressed, it makes it easier for you to have a conversation with your bioinformatician or whoever's making those final figures for you about what you're looking for and how to generate them. So that's really how I see Loop Browser. There's also the ability to do some cell annotation that we'll show you guys today as well. Okay. So what I've done, again, cooking class style, is I've downloaded all of these files ahead of time so that it's a little bit easier and quicker for us to look through them. So what I'm going to pull up first is our web summary HTML files. So just like we were looking at in the cloud, there's going to be one file for each of the samples in that multiplex library. Let me zoom in so this is a little easier for everybody to see, much better. Okay, so anytime we have a web summary QC report, and we're running Flex, specifically the multiplex version, you'll see Cell Ranger Multi at the top. This is just telling you what version of the pipeline you were running. Bless you. More information down here, right, about the pipeline version, the name you gave it for the run ID, and then the sample ID that you provided. So this is just more a sanity check that what we processed is exactly what we thought it was, and you didn't accidentally download the wrong file. Right, we see this nice green box at the top. No critical issues with a check mark. That's a great place to start, right? That's letting us know that there weren't any glaringly obvious things about this data that were problematic. Okay, underneath here, there are 3 tabs. Again, this is going to be specific for any time we're doing a multiplex Flex library. There's going to be a gene expression library tab. That's your overall library. That's all of the samples that you've pulled together. All of the metrics across that library without them being demultiplexed are going to be in this tab. We have our sample tab, which is going to be specific to the sample ID that's at the top of the document. So here, this is going to be all of the metrics specific to our I sample. And then we have this experimental design tab all the way on the right. I really like the experimental design, especially as someone who's often helping you guys troubleshoot your experiments. It's a really great way to understand how this experiment was set up, especially if you're just the person who kind of got the data dumped on you, which sometimes happens, right? You might not have been the one to design the experiment. Makes it very simple and straightforward for you to understand how this was set up at the bench, so that way you can make sure that you're looking at the data properly. Okay, so going back to the overall library QC, there are a couple different tables as we start to scroll down. We have this cell calling quality table. Again, it's not going to be as informative as the per sample, so we kind of just take it at face value and we're looking at the overall library. We can see that we've called 46,000 cells. I know based off of what's on the public data set website that this was targeting originally 40,000, so we're right in that ballpark of what we're looking to see. Confidently mapped reads and cells, right? As you can see, when you're using the HTML version of these files, a lot of the different tables are interactive, which I find super helpful. There are a ton of different metrics. You're never going to remember what all of them mean, right? So being able to hover over the top and just get a quick description of what you're looking at for each metric is really useful. So here what we're looking at is reads that had valid 10x barcodes, so barcodes that we know should be from our gel beads, a valid UMI sequence, which is essentially tagging the individual transcript or probe pair we picked up, and then something that was confidently mapped to our alignment files or reference files. So 96% is definitely what we should be seeing, especially with Flex where we're dealing with a probe set, right? There shouldn't really be a lot of ambient RNA that's getting picked up that would map to your specific reference file. So I would expect this metric to be particularly high when we're dealing with Flex data. And then over here, we have this initial cell barcodes passing high occupancy gem filtering. This is going to be more specific, again, to multiplexing with Flex. Like I mentioned before, we've now launched a new version of Flex where customers will actually be able to plex up to 384 samples into one library. So as you can imagine, we can really kind of increase the throughput there. So we just want to make sure that we aren't overloading the single cell partitioning system. For something like a 4-plex, I would expect it to be at 100%. As people get really, really high in plexity, this number might not be at exactly 100%. If you're running a single plex or a 4-plex and you are seeing some numbers go down here, it could be an issue with the gem formation, the actual partitioning of the cells themselves. Okay, next up we have a barcode rank plot. again, we're gonna have a version of this for each individual sample, but I'm just gonna take a minute here to talk about what this plot is because it's actually really useful for you guys in understanding the quality of your samples. So, like the title is letting us know, all we've done is we've ranked all of the 10x barcodes that are in your data, so those cell barcodes from our gel bead oligos. The ones that have the highest number of UMIs are going to be closest to the y-axis, and the barcodes that have the fewest number of UMIs are going to be furthest away from the y-axis. What are we looking at here? As a byproduct of the way we partition the cells on our chips, we expect to have empty droplets where we should see background noise or little to no probe pairs mapping back to those droplets. And so by ranking all of our 10x barcodes, we can essentially use an algorithm to differentiate where we are cutting off from droplets that did contain cells to empty droplets that maybe just picked up a few probe pairs and have a low number of molecules that were converted into our library. And so that's kind of what we're seeing here on the coloring of this plot. So you can see that for these really high UMI count droplets, we are seeing that the barcode or the plot line is highlighted in a dark blue color. And as we start to go farther down that line, this is the other reason I really like the HTML. It's really interactive for the barcode rank plot. We can see that we start to get what we refer to as this sort of cliff or this plateau where we're dropping off on the UMI content. Totally normal. We expect that with the way we're partitioning our cells. And so it's somewhere along that cliff where our algorithm is going to try and differentiate between what is a real cell and what is essentially in this background noise population that we have down here. And so if we wanted to, my cursor, you can actually highlight over any region on this plot and zoom in if you wanted to get more information. Again, this is just a sanity check to make sure that you're capturing the number of cells that you thought. It lets you know if there's a lot of background noise in your samples. Generally for Flex, we should see less background noise, but you always want to check, right? And so here we can see where our algorithm went from calling cells to deciding that it's background noise, right? So the 48,434 droplet that it came across, it decided this one's background and everything after it is background as well. So barcode rank plots are really useful. They're a really good way to get a sense for how your emulsion was generated at the bench and gives you a little bit of information about the quality of your sample as well. Okay, mapping quality, again, this is mainly going to be centered around the probes. So we expect these numbers to be really high, assuming that you're using the right reference for the probe set that you utilize and that your samples were able to hybridize to those probes efficiently. We should see high numbers for these first 3 metrics. And then for this last 2 metrics here, we should see very low numbers. Basically what we're looking for here is non-specific probe ligation for the split mapped probe set metric here. So we shouldn't be seeing the left-hand side probe from beta actin and the right-hand side probe from GAPDH ligating together, right? They should be ligated to their actual true probe pair, and so that's what this metric is telling us. Generally, if this is higher, it has to do with a lot of unbound probe being left over that allows for sort of spurious ligation events to occur. And then over here, half map probe set, again, this just has to do with washing away those unbound probes so that we don't have a lot of them left over to be picked up in your final library, so these numbers should both be equally low. Sequencing quality, right, is going to have more to do with the sequencing run itself. We should be seeing high metrics here. If there are issues with the sequencing quality, we can always rule out whether or not it was like a sample or library prep issue, but it could also just be that something went wrong with the sequencing run itself. And then lastly, what I'll touch on here is this metrics per probe barcode. This is just a good way at a quick glance to see how your library ended up demultiplexing. I was just talking about this with Yuri earlier, but when we're pooling those samples together and washing them, we're going to get a little bit of variability in the recovery just by the stochastic nature of the cell loss during those washes. And so like we talked about before, we're anticipating capturing around 10,000 cells per sample, and we're pretty much more or less in the ballpark for all 4 of our samples. So that's a good sanity check here that things went well. So now, yeah, Nelson, what's up?
These probes to the transcriptome are targeting pre-messenger RNAs or mature messenger RNAs?
We target exons.
Just exons.
Yeah, so that it would capture multiple.
Does it also capture, does it include exons to the mitochondrial genes? Will it give a readout on how much mitochondrial RNA is in the sample as a reflection of how stressed the cell association is? Because that's what you would get in a typical single-seq sequencing.
Yep. So we do have what we call like sample QC genes that we target, mitochondrial. I think there might even be a couple ribosomal as well, just to make sure that that's not the main thing that we're capturing out of your samples. So yes, those are included. And like
ribosomal, that's ribosomal RNA or ribosomal mRNAs, ribosomal protein?
I'd have to go back and double check. I think it's mostly mitochondrial, but I can get you a firm answer. But like I said, the CSV files are free to download off of our website. We have no secrets about what's in that probe set. So if there's something that you want to make sure is there before you get started, you can always check. And then we do have some documentation for generating your own custom probes. So this has come up a lot with other groups as well. Like we were talking about before, maybe you're looking at an alternative splice site, a deletion. You can design a probe that will specifically target that. We try to avoid all of those things with the probes we designed, just so it would be more of a catch-all, looking for the presence or absence of those transcripts. Yeah. Okay. So we talked about the sample the overall library QC. I just want to touch on the sample. A lot of the same metrics you'll see on the sample page, right? All of the cell calling quality metrics you see here will be specific to that individual sample, right? So for our eye sample, we captured about 14,000 cells. We see we have good mapping metrics, which is what I would expect. Median genes per cell, median UMIs per cell. These are going to be a little bit more variable depending on your sample, right? Excuse me. There are going to be certain cell types that are going to express a much larger suite of genes than other cell types. There are certain cell types that just have a lot more RNA content than others. So these metrics can definitely change depending on the sample that you're working with. So try not to get too hung up on what you're seeing there. We have the total genes detected, and then we have the total number of reads, right? So we sequence the whole library, and this is just telling us from that library how many reads were coming from this particular sample. If we wanted to do some quick math, which I did ahead of time with a calculator because I don't trust myself, you would see that the number of reads per cell, which is an important metric for us to make sure we've sequenced enough to capture the complexity of your library, is about 22,000 read pairs per cell. So we just take this sequencing number here and we divide it by the number of cells, and it gives you about 22,000. So this is a really deeply sequenced library. We generally recommend 10,000 read pairs per cell. So you're definitely getting the full complexity here. Barcode rank plot looks very similar to the one that I just showed you guys. The major difference here, right, is that the number of barcodes we're looking at is about a quarter of the previous plot because we're just looking at the subset of eye cells that we captured. The last thing that I'll point out down here at the bottom is we do have a UMAP projection at the bottom. It's not going to be sort of where you want to land to do like you're really interrogating into your sample, but it's a good quick glance. What I like to look at here, especially to our conversation about like lower quality cells, and this would apply not just for Flex, but also for your RT-based capture. So there are two different versions of these UMAP projections, right? So for UMAP projections, for folks who don't know, we're essentially clustering cells based off of the genes that we detected in these libraries. So dots that are really close together or clustered together, like the ones in this one, are most likely a similar cell type. They have similar transcriptional profiles. So we have two different versions of these projections. On the left, we are coloring the different cells based off of how many individual UMIs those cells have. And then on the right, we're coloring them based off of the algorithm we use for clustering cells. So again, we're using the UMAP algorithm here, and so it's saying that everything highlighted here in red for cluster 14 is most likely the same cell type, right, based off of that algorithm. So for a sanity check, what you can do is look for really cool blue clusters here, so things that are really low in UMI counts. And so we can see here this little purple guy, for example, which is cluster 15, is pretty dark blue, so it has a low number of UMI counts. So if I wanted to double check to Nelson's question, like what is the highest expressed gene in that cluster, so that I could rule out whether maybe that's just background noise or dead or dying cells, I can just scroll over to cluster 15. And then I'm just gonna toggle on the log2 fold change column, and that will allow me to sort the data in this table based off of what's the most highly expressed in this cluster in particular. So then I can scroll all the way back over. Like I said, it's not super user-friendly in the web summary, and then just do a sanity check that the top 10, or we can even make this a little bit longer and do the top 20 rows, that what we're seeing for the genes that are coming up aren't those mitochondrial genes that we have for sample QC, which I'm not seeing here, right? So this is probably just an indication that these are lower RNA content cells. They aren't necessarily dead or dying. They could still be biologically relevant, right? So web summaries are a sanity check, right? They're not the end all be all of where we're going to do all of our downstream analysis. This is just getting a feel for what it looks like. The other thing I want to point out, and the reason I reran this data for you guys, is that with the newer versions of Cell Ranger, we have this auto cell annotation function. So I'm going to click over to this tab and just show you what it looks like. So this is something that's still in beta test. So we've developed it with a community group that has written the algorithm. It's been trained on healthy cells mainly, so it might not be the best annotation for your particular sample type. You kind of need to go in knowing that this is fully a beta test, and I'm going to show you some examples of that today. What I like about it, and what we're going to use today in the example for kind of showing you how to use Loop Browser, is it ranks the different cell type compositions that are in that particular sample type. So let me scroll up so it's a little bit easier for you guys to see, right? So for this, we're talking about our eye sample. The top type of samples or cells that we were able to annotate using this algorithm were neurons. I'm not an eye biologist, but to me, this seems pretty on par with what I would expect coming out of this tissue type. If we go to the right over here, we have this subtype table. So within the neuron group, that algorithm was able to further define these 4 additional cell types within that sort of neuron class of cells. And so we can see here retinal rod cell is coming up as the most prevalent in that neuron group. Again, this is biology is tracking for me. I'm definitely not an eye expert, but this is definitely giving me a good sanity check that this annotation is probably going to be helpful for me. I always tell customers, like, don't take it all at face value, especially if you're an eye biologist or you work on retinas. You're gonna know what that cell type looks better probably than this algorithm, but I think it does a good cursory job of getting us started. So now what I'm gonna do is we're gonna switch to Loop Browser, which I will show you guys how to find Loop Browser on our support website. And then I will show you the loop files that I downloaded, and we can kind of go through them together. So we're on the support website. There are a couple different ways that we can get there, but I'm going to go for the most direct, which is just scrolling down to software support. And you can see we have a couple different versions of our different Cell Ranger pipelines and Loop Browser. Today we're going to focus on this version of Loop Browser, and then you can download depending on what version you want and whether or not you're using Mac or PC. There are going to be different options. Hopefully I didn't accidentally start a download. But this is where you would go to find it, right? So from here you have the option for Windows or Mac. Obviously I've already downloaded this, so we're going to pop out of this window. I'm going to get rid of our web summaries, and we're going to start by looking at eye in Loop Browser. Okay. So first things first, just kind of a primer for Loop Browser for folks who have never used it before. Similar to what we saw in our web summary, the main window here is going to have, instead of UMAP, it's a t-SNE plot. So it's just a different algorithm that is accomplishing that same cluster generation, trying to group your cell or your samples together. If you look across the top here, right, we have kind of our standard save button. We also have this projection type button. You can see that it's going to default to t-SNE, but there is a drop down, so you can switch to UMAP. Quite frankly, I think it's more personal preference and kind of depends on the data. Sometimes UMAP does like a better job, I think, kind of separating out your clusters so they're easier to work with. Really doesn't matter. It's just personal preference and what you guys are interested in. We also have this thing called split view. I really like the split view. I think it's a good way for you to break up what you're seeing on your t-SNE plot. There are different ways that we can split. So we can split based off graph-based, and that is just going to use these clusters that were sort of default determined in our clustering algorithm. So if I were to select that, it's just going to break out all of those different colored clusters that we just saw into their own separate boxes. It's really nice if you want to take a closer look at one cluster in particular and figure out what's going on. for today, because I really want to highlight the cell annotation, I'm going to scroll down. We also have these k-means options. It's essentially asking the data to be forced into a certain number of groups, right? So if I said, I realize graph-based, you think there are 18, 19 different clusters, but I want you to force the data into 2 clusters. What would that look like? Or I want you to force the data into 7 clusters, right? And so on and so forth. So you can really play around in Loop Browser without having to be super bioinformatics savvy, which I love. For our example today, what I'm going to focus on is our cell type annotation, that beta version that we're using. So this allows me to break out into all the different groups. For anybody who's ever worked with like Adobe or anything like that, we have kind of similar tools up here in the corner. We have this lasso tool, which allows you to highlight things. The pan tool is just allows you to kind of click and drag around. It also allows you to zoom in and out. I should have brought a regular mouse for this, but that's okay. And so that makes it really easy if you want to zoom in on a particular cluster, which is what I'm hoping to do here with our example. So what my goal here is to highlight for you guys in Loop Browser is just how you can play around in it, and hopefully show that the annotation tool does a decent job for some sample types. Okay, so to that end, let's start by digging into the neuron group. So I should zoom back out a little bit and find where my neurons went. And probably right in front of my face, and I just can't see it. Ah, here we go. So this is our neuron population of cells that our algorithm called. Now all I want to do is just a sanity check, right? We saw that retinal rod cells were up at the top of the subtypes that the algorithm was able to identify. So I'm just going to test that a little bit. So one way that you can go in and look for different features or genes in your data is by clicking on this features tab on the right or on the left hand side. There are different things that you can do over here. We won't have time to talk about all of them today, but really clusters and features I think are the main ways that you can use Loop Browser. So if I click over to features, now what I've done is I've removed all the colors from that pseudo coloring from the t-SNE projection, and now I have the option of putting in a gene or feature in here. If you already know the sample or the cell type that you're looking for, and you have a really well-defined list of genes that you are expecting that cell type to express, you could generate a CSV file of that ahead of time. And so to kind of show you what that would look like, if it's going to pull this up for me. Oh, no, that's not what I wanted to do. Right, so we have the option of uploading a CSV file, which is essentially just a version of an Excel document where you'll have the gene name, like the ensemble ID, and then what you want to group it as, so T cell, neuron. I can pull up an example after we're done today, but essentially you could make that ahead of time to annotate your cells. Here, what I'm going to do is I'm going to delete these untitled lists that I accidentally just made because I don't need them. And I'm just going to search for one of my retinal rod cell genes that I know should be expressed and that I'm hoping will show up in this neuron cluster. So obviously I did my homework ahead of time and I was looking into some genes. So we're going to try NR2E3, which should be a marker for our retinal rod cells. So what we're seeing here is good for a sanity check, but let's talk about what the pseudo coloring is that you're seeing here. So up on the right hand side, we have this scale value of expression. That's really what we're looking at. It's a heat map of expression. The lighter in color it is, the less that particular cell or droplet was expressing that particular transcript. The darker it is, the more expression we captured for that particular cell. And so this jives to me, right? I wouldn't expect all of the cells in this cluster to be heavily expressing this transcript, if not all of them were identified as retinal rod cells. So this gives me sanity that what we're seeing in our cell annotation is real and is likely accurately calling these retinal rod cells, right? Another example, so if we were to really quickly here pop out of this, we can go back to our annotation, or excuse me, our web summary, and just look at what some of those other potential cell types were. So for I in particular, right, the next big one here is squamous epithelial cell. If I click on this, it'll bring up that subtype. And so we should be seeing keratinocytes, and they should be pretty heavily expressed in this population. So we'll do one more example with this one, and then I'm gonna switch over to stomach because I think it kind of of shows a different version of the cell annotation. So for the keratinocytes, let's go full screen, make it a little easier for you guys to see. Again, I've done a little homework, and I know that they should be expressing KRT12. So good news is we're not seeing that in our neurons, right? Which is great. We're hoping to capture this in a different population of cells. And that is what we're seeing over here, right? We saw in our web summary that the squamous epithelial cell population, about half of them should be expressing or should be these keratinocytes. And I'm seeing a lot of really dark red dots here, which is leading me to believe that this annotation is doing pretty well for this cell type. Okay, so I know we're coming up, we've only got a couple minutes left, so I just really want to show you an example of what stomach looks like, because I think on the opposite end of the annotation spectrum, it shows that it's not great for every sample type, right? So if we were to go into our stomach web summary, and we'll just pull up the cell annotation really quickly, what we're seeing is that we really just have this one major type of cells. Zoom in so it's a little easier, and it's epithelial, right? I did a little Googling and tried to read up on stomach, and this kind of jives, right? This should be, there are a lot of different types of epithelial cells that should be expressed out of the stomach. Most of them are expressing some kind of like mucosal genes and are expressing mucus themselves, but there should be a lot of really specific cell types to the stomach. What's concerning, right, is when I look over at this subtype, I'm not really seeing anything that is ringing bells for me as far as stomach should go, right? So I'm seeing pancreatic, I'm seeing urethra, luminal epithelial, that's mammary gland, and bladder. So something is up. Either I ran the wrong sample and I labeled it improperly, or maybe this algorithm is just not well suited for stomach. So a good way to check that is to just go into Loop Browser and look at the stomach data set. So let's do that really quickly. All right, so same deal as what we did with eye. I'm just gonna split on those cell types, right? We know that about 90% of these cells are falling in that epithelial group. So I'm just gonna zoom in on that, and then we're gonna look for some of the features. Again, things that I looked for, so kind of the major classes that I was able to find. So we have these, what are they called here? Mucus neck cells and progenitor cells. Not a great name, but that's what they've named them. And they should be highly expressing ALPI. I'm seeing some expression here, right? If I were to zoom out and make sure, I'm not seeing in other groups, which is great, but there is a large proportion of this cluster that's just not lighting up with anything. Another major group should be just gastric mucus cells, right? So those are the mature mucus secreting cells in the stomach. So for those, we should see expression of, this one's showing up a little bit better, right? But there are still, if we were to zoom in here, there are still a lot of cells or dots in this particular cluster where I'm not seeing either of these markers lighting up. If I wanted to look at them together, I could do that. So we can press this combine all button here, and this is showing us the co-expression of both of those genes within this particular subset of cells. And we're seeing that there are a lot of cells back here that are not lighting up as either. So to me, this is an indication that maybe for stomach cells in particular, the algorithm that we're using isn't really able to drill down with enough granularity for the different epithelial cell types that are in this tissue. So, you know, it's still worth going through Loop Browser, trying the annotation, the auto annotation, right? It does it for you, so you don't have to do the bulk of the work. But the caveat here in the life lesson is that you guys are ultimately going to know the biology of your samples better, right, than Loop Browser or the annotation. So make sure you're double checking that the algorithm is really behaving as it should and it's making the right calls. You guys can always re-annotate your cells using Loop Browser, right? So if I wanted to pull out just the ones that are positive for one of these markers, I can use my lasso tool to highlight over them, and then I can rename them as a certain cell type, subtype, and then export that, right, and use that for my downstream annotation. So I know that was super quick, and I know I threw a lot at you guys, but hopefully it was just enough to get you guys interested and excited in using Flex and playing around with the data. We can send a follow-up with links to the public data that I pulled this from and ran. So if you guys wanted to run it on your own, you're more than welcome to. There are also a lot of different public data sets that are on our website. If you want to download one that's a little bit more relevant to your project or your research, feel free, right? You can use the cloud for free to analyze that data, and then you can really get a sense of what it looks like ahead of even performing one of your own experiments. So with that, I'd be happy to take any questions you guys have.
Yeah. One question of, do you have a comparison from these tissues that are presumably fresh versus fixed FFPE?
Yeah, so these were frozen tissues that were run through that chop fix protocol that I mentioned. We do have some FFPE data sets on our website as well. I just picked chop fix because it seems a little bit more relevant for the researchers that I've talked to on this campus. It seems like a lot of people are getting frozen tissue. But yeah, we have a lot of FFPE data on our website, and we'll be sure to include the links to that in the follow-up. Yeah. Any other questions?
Any other questions for Juliana?
All right.
Thanks so much, Juliana. And just remember the deadline for the Flex grant is November 16th, and please reach out to Yuri, Juliana, myself, we're here to support you, and good luck.
Thank you everyone for your time.
Symposium, November 14th, Friday. Okay, thanks.
Thanks, guys.
Thank you. That's great.
Yeah, right? Yeah, this is a good way to highlight it. Yeah, and like I said, it's when we
There's also a document that's like resources.
What? Oh, I haven't yet, no. Hi! Awesome.
Just for today,
Right. Yeah.
Yeah, so over from ballpark and see
Yes. Yep. Yep. So we can talk about it on Friday, and I will send you a document on our website that walks you through the and then you would order them from a primer from like IDT or Twist, and then you just select them into the probe set. Super easy, super straightforward, but we can definitely talk more about the probe design and just making sure that you're feeling confident. That's very easy.
Yeah, I was going to
Okay. Yeah.
Sounds like human probe has had like a session data from
Yeah.
We just want to learn more about like your experience on
What we've done, so we haven't done any like official tests internally, obviously just because it's hard to get access to those types of samples. But what we've done is we've done computational prediction, so our AB or basically what they think is most likely to overlap and not overlap. So we'll set with different, like the most common